with that. Good afternoon, everybody. Good afternoon, everybody on Zoom. Welcome to this seminar. Today we have Dr. Pankaj Kumar. He's an adjunct professor at Miami Dade College, teaching general biology and plant biology with over a quarter of century of research experience working across various countries in Asia and now in the USA. He focuses on taxonomy, ecology, and conservation of native orchids. Please applaud and
Thank you so much. Thank you, Professor Lin, for organizing this, and Professor Hong Liu for giving this idea to come here for presentation. So today I'm going to talk about one endemic North American orchid, Cypripedium acaule. So my wife is sitting here. We kind of have been moving from Texas to Florida to New York, up and down a lot of times. And whenever I get time, I try to study orchids either there or here. So this is one of the species which I started my study on. And the presentation today is very formal. It is based on 1 year of data, 1 year of observation, and it kind of opens up some research questions for which I'm giving some hypotheses, and then it can be carried forward either by me or in collaboration with some of you, or if some of the students are interested in this work. So I'm going to talk on the pollination ecology of North American endemic moccasin orchid. Moccasin means a leather shoe. So there is a legend that the local native North American girl was looking for some medicinal herbs, and it was snowing outside, and she walked on the snow, cold snow, with snowing weather to reach out to some of the herbal plants in the forest to bring it back for the tribal members who were suffering. And wherever she stepped, this moccasin orchid, this grew there. This is very interesting because something similar you find in Hindu scriptures in India, where Lord Krishna is said that when he started walking, on each step, the lotus flower bloomed. So I'm going to talk about this orchid. I've been looking at a lot of aspects which are available already on this orchid, and I realized that it has many informations which are either missing or something which I do not agree to. So why do I want to study? Because I want to have a conservation plan on this species. When you have conservation plan, what does it mean? It means we are talking about ecosystem stability. We are talking about conservation or protection of biodiversity. We are talking about how we can access the habitat loss with respect to climate change. And we are also not in this, but we are also talking about how to assess the human benefit. A lot of funding agencies nowadays, they are looking at the human aspect of any project. So if your project has something which brings about benefit to the society, the likelihood of getting it funded is high. And then we have the cultural aspect, what I told about the Native American girl, wherever she walked, this orchid, they appeared on the ground. Then for places like USA, where there is not much diversity of animals, there are, but not large animals, some species like this can be taken as a flagship species, because if you are going to protect this species, you are not just conserving this, but you are conserving the whole habitat and any other organism which is living in this habitat. One of the best examples can come from England, where there was Cypripedium calceolus. It is common in Europe, but it was supposed to be extinct in England somewhere around early 19th century. Then in 1930s, someone found one small population in the outskirts of England in Yorkshire, and he kept the location secret from everyone. His kind of family members used to go there. Then some nursery owners started to go there, and gradually, when the government in England they came to know about this orchid, they appointed a guard, especially to look after this orchid. In 2018, they kind of propagated this species and they relocated them back in the wild. So why not in USA? Why you cannot do some things like that in USA? There are chances, there are species like this, not just this. Cypripedium has around 13 species in USA which are native. Some methods, there are a lot of methods, some of which I am using during my study. So those are underlined. The topic highlight is underlined. So first, I was looking at a lot of literature, whatever I could find on this species. And I was looking at GBIF, I was looking at iNaturalist to go to the sites where this orchid is found. So I'm lucky that my wife was taking me to every site wherever this is found, and we could kind of monitor if the plant was still existing or not. So we went to a lot of sites and we found at least 5 different sites. And one of the sites where I did this study, it has at least 300 species above ground last year. So there should be more which didn't emerge out, and it will, like sometimes some species, they come out of the ground alternate years. Then I looked at the habitat integrity based on literature and based on the habitat where we were surveying the preferred habitat, and if there is no threat to the habitat itself. If there is threat to the habitat, you cannot conserve the orchid. And lots of orchid, not just in USA, but in other parts of the world are exploited. They are collected, and that is one thing, but the habitat is also disturbed. They are constructing new houses, the roads, and playgrounds in forested areas. These lead to the loss of habitat, which is needed for this species. Then I focused a lot on the pollination studies. I love looking at plant-animal interaction, so I focused on the pollination of this species. Everyone knows that it is pollinated by bumblebees, so bumblebee queens. So we'll talk about that later on, because if the pollinator is not there, you cannot help in the survival of this species. Then there are aspects of genetic studies on that. I do not do genetics myself, but I have options what can be studied on this species. And then once I have this data, which needs to be compiled for analysis and then communication. And once it is done, I wish to do the conservation assessment, review the conservation assessment. I'm also the Red List coordinator for Orchids of the World, only one person. So I do a lot of conservation assessments of orchids across the world. And then when we have all those informations, all those data, we can come with a species action plan for this particular species. And then this study can be replicated on another species which are found in this region. So what do we know about this species? Taxonomy, we know taxonomy is very straightforward. There is distribution. I looked at a lot of literature and I found a lot of discrepancy in the distribution data of this species. Then conservation assessment, we'll talk about it. It has been assessed as least concerned in 2014 by Ranko and Sharma. Pollination biology, people have known that this species is pollinated by bumblebees, queen of bumblebees. And I have observed the queen bumblebees pollinating this species, but no one knows anything more than that, how the pollination occurs, what happens, why it happens, and is it impacting the population size? Is it helping to grow, for the species population to expand? Or it is not helping, we will talk about that. Then again, population genetics, this species comes in three colors. One is this one, and then there is a white form, and then there is a very dark pink form. So, so far in New York, I have not seen the other two forms. So I have to go out of the state to look for other forms. Then the effect of climate change. This species is a spring flowering species. This grows in areas where there is snow during winter. So how is this species going to be affected if there is no snow, if the winter temperature is higher? So we'll talk about that also. Looking at taxonomy, taxonomy is very straightforward. The plant was collected somewhere in 1786 from North America. No one knows from where exactly from, but it was collected and sent to England. There the plant was growing for 3 years, and 1788, the plant was described and published in 1789. And this belongs to the family Orchidaceae, of course. And Orchidaceae has 5 different subfamilies. It belongs to the subfamily Cypripedioideae. It likes to grow in dry or wet forests, bogs, brushy barrens, heath, roadside, on high acidic soil. And the elevation range is very large, from 0 meter ground level to almost 1,200 meters. This information is based on flora of North America. So this is the specimen which was collected from North America. We don't know from where, and it was preserved in 1788. 1786, it was collected and it was grown in the nursery in England. And that specimen is at British Museum now. So if you look at the Cypripediums, they are called Cypripediums because the lip is pouch-like, the lady slipper-like. And so Cypripediums have, it's okay. Cypripediums have 2 types of opening. This is the opening which is present here. This is called orifice. This structure is the modified lip. It forms a pouch-like structure. In some of the species, the pouch opens on the top. So this is the opening on the top. Then in other species, only one in USA, where the pouch opens on the front. But what happens is the upper rim is kind of folded inside. It goes inside, doesn't touch the back wall, and it forms a furrow on the front. So the orifice is here. The importance of orifice is for the pollinators, because pollinators are going to enter through this space, whatever is present here. And this is folded, and it has a margin inside, which we will look at later on. If we look at the morphology of the species, it's called Cypripedium acaule. Acaule means without stem. There is no stem as such or above ground, but the stem is very short and very much underground, and it has usually two leaves. But I've seen plants with single leaf. I have never seen plants with more than 2 leaves so far. And it has single inflorescence with a single solitary flower at the top. You can see the height of the flower is lower than the height of the fruit. Can any student make a guess why? It's okay to talk if you want. The height, this is a mature open flower, and this is a fruit. So it is pollinated by a very specific insect, which I will talk about. So one of the reasons is when it rises up, it can disperse the seed much further than if it is on the lower height from the ground. So it after pollinated, it kind of still grows a bit and rises up above the ground. So this is the fruit, they are leaves. This is the flower here. The flower, if you look on the top, is the floral bract. Inside is the dorsal sepal. So all orchids are monocot. It is a trimerous flower, 3 petals, 3 sepals. So here is the dorsal sepal, and these two are the, dorsal sepal is here, and on the back are the 2 lateral sepals, but the lateral sepals are fused together to form something called synsepal. It looks like one, but it is a fusion of two lateral sepals. Then here are the petals, one petal, two petal, third petal is modified to form a pouch-like structure. On the back, here is the staminode, which is the which is formed by the fusion of male and female organs of the flower. And on the back of the staminode, you can see the yellow things. Those are two anthers placed on the back of the staminode. You cannot see here, but below the stamen, there is stigma, just below there. It is basically meant for the purpose that when insect goes in from here, and it comes out from there, it is going to touch the stigma first. So it avoids self-pollination. It is going to bring pollen from some flower. It will go inside and put on the stigma. Then it will take the pollen and come out. We'll see that happening actually later on. So let's talk about conservation assessment, which I said that it looks a bit not correct to me. It was assessed as least concern by Ranko and Sharma 2014. However, they listed a lot of threats. Still, they assessed this species as least concerned. So some of the threats were invasive exotic species. There are two species. Lonicera japonica, Japanese honeysuckle, and Rosa multiflora. These two non-native plants, they occupy the habitat and they kind of suppress the growth of the orchid. Then there is urbanization and real estate development. A lot of the parts in upper USA are being developed for real estate. The plant is collected for horticulture. People like to go and collect the plant, grow in their garden, kill it, go back again, collect more plants. So that's a free plant coming, but it leads to the depletion in population. Plant is also collected for medicinal use, and I have also observed herbivory. The leaf is eaten by some animals. Sorry, let me drink some water. So they actually talked about continuing decline in population size, and they talked about continuing decline in quality of habitat, one of the two criteria which were used to assess this species as least concerned. When you say least concerned, it means it has no priority for conservation. But when we look at the species distribution range, when you see here, this is from flora of North America. The species distribution range, you have to focus here on northern Canada. There are some parts where the species was supposed to be found. There are some parts here where the species was supposed to be found. This is from flora of North America. And that area is of concern because when we look at NatureServe's data, latest data, they don't mention about the occurrence of this species there or there. And the species is known to be vulnerable in some parts of Canada, and some parts they are actually critically imperiled in USA. There are some data missing from some parts. I was also told that it was reported from some forests in Texas, but I couldn't find the specimen, neither I could find an authentic reference. So I'm not sure if it was there, but there are some forests, good, similar looking forests and habitat which are available in Texas. So there was a chance, and there are parts in Texas where you get snow, so chances are there. However, I don't have any proof right now. So we are based in Brooklyn, and we also lived in this part of Long Island in New York. So that was the closest area where we could search for this species. We used to take a ride, go out in the wild looking for the plant in the season, and we have traveled from here to here. It is almost 90 kilometers, 90 miles or 90 kilometers, I forgot. I'm Indian, we talk in miles, we talk in kilometers, so I always get confused about miles. So, there is a reference recently came out last year on the orchids of Long Island by Lamont, and he claims that the orchids are not found at least 4 localities. It has been extinct from at least 4 localities on Long Island. He was focusing on Long Island. The number of mature individuals have gone down. It has extirpated from the Queens area. We have done a lot of survey here, and we have done and we have been to this part. So we have done all the surveys here. This part, there are some forest, and this part. At this part, there is no forest left. So you can see that the species has declined. There are reports of the species to have declined in various parts of USA. This is the data from the Canadian government. So if you look at 2000 data, the species was imperiled here. However, in 2005, it was found here. It was found here, and then it got imperiled again, and it was considered vulnerable. You can see that there is uncertain data from this part, but ultimately there is no data from that part, the upper part, and there is no data from this part. So species has declined a lot in Canada. We do not have data for USA. There is also decline in the population of the pollinator, which is very important, because if the pollinator is not there, the species survival means nothing. It cannot persist on its own. And hence, I think at least this species qualifies for near threatened, or if we have good data, this will certainly go under vulnerable. So let's look at pollination. This is the structure of flower we talked about earlier. It has unique adaptations for pollination. There is availability of pollinator when the flower is open. It is pollinated by bumblebee queen, Bombus vagans, as per some references. The flower is brightly pink color, which is very attractive to bumblebees. It has color, it has fragrance, it has a very strong fragrance. However, it has no floral reward. Duration of anthesis is very long. It can stay open up to 3 weeks if it is not pollinated. If it is pollinated, it will shrivel fast. So about the pollinator, we know the queen bumblebees are the pollinator. These bumblebees, they hibernate under the ground in the forest, and they hibernate during the winter period. After the snow has melted, the insects try to come out. They are super hungry, and they are looking for food. And the insect doesn't fly more than 2 meters above ground. So it will fly much lower than on the ground. And actually, my data says that it didn't even, my insects, I had at least 5 complete data where my insect never went up above 1 meter. So it is there at a very low height, and that's the height where you can see the flower. Even if it is 1 meter high, you can see the flower, and the insect is facing the front of the flower. That is exactly where it wants to go, the red color, and then it has fragrance. And the lifespan of Bombus vagans is 1 year. This is a very important point. If you remember it, we will talk about it towards the end of this presentation. So we will look at the phenological timing. The plant, it is going to stay underground for the whole winter. The plant is going to lose all its leaf. There is underground stem where it stays there for whole winter till the snow has melted. So once the snow has melted, the plant kind of starts to emerge. It is slow, but you can see the first appearance of the leaf buds and the flower buds. Because it is on the ground, it is not easy to see, but if you know where the plants are, you go there again, you can find. And then it gradually becomes healthier and healthier. And by this time, the plant, the leaves are fully expanded and it is enjoying the sun. So it is growing under the shade. And during winter, the trees have lost the leaves. So during this time, there is enough sun and it is enjoying under the sun, under the tree, making a lot of food. How about flower? Flower and leaf, they kind of emerge during the same time, but the flower is in the bud condition. The flower stays in the, it keeps growing, and by around this period, May, the flower is fully open and it stays open up to 3 weeks if it is not pollinated. Looking at the pollinator also goes through a hibernation. So in winter, it is going to go underground and stay underground for the whole winter until the snow starts to melt. So somewhere around here, it starts to come out. And as I said, it is hungry, looking for food. And so it is the main aim for her is a female. So main aim for her is to gather as much food as she can. Looking at the temperature during the winter, of course, it is very cold. By this time, it is getting to get warmer, but during May, it is not too warm, not too hot. If it is too hot, the flowers are going to dry. It is not too hot, not too cold. So if we look at the timing matches. Timing matches in the sense in May, the plant is in full health, the leaf is fully expanded, the plant has flower, which is fully open. There is availability of the pollinator and the weather is not so hot. So if, as I said, if it is too hot, the flowers will dry up very fast. So, and once the flower is pollinated, it stays almost for 3 months to get the seeds mature. Then one more interesting thing is what happens to the seeds then. The seeds, once dispersed, they also undergo snow period. So they remain buried under the snow. And of course, they will not germinate during the snow. They will germinate somewhere after the snow has melted, somewhere here. But we have not studied that part yet. So the main question is, I'm trying to coin some questions from this study. One of the question is, what triggers the flowering? And how will the species respond if the climate changes, if there is no snow, if it becomes too hot? If it becomes too hot, we know that the flower is not going to survive for long. But what if there is no snow? When there is no snow, a lot of things will be affected, which we will talk later on. So here is a video which I actually compiled based on images, and it is a 3-minute video based on the exact 2-minute video based on the exact timing of the event. So you can see the insect is on the pouch, and insect knows that there is something. Why it knows? Because it has a fragrance. It can sense the fragrance. It doesn't know how to go towards it. So the insect spends almost 1 minute looking for the entrance, how to go inside the flower. And this insect has a brain. It has a kind of 3 millimeter cube brain with around 1 million neurons. So it has a brain which it uses. And first it is going in front, and then it turns to the side because it knows that it cannot enter the pouch if it is facing towards the flower. When it turns the side, it is inside. Now it is trapped and it is looking, it is already deceived. There is no reward. So now the insect, she is a queen, so she is deceived and now she is frustrated. She cannot come out. The only way is she can see that there is a window there. So she has to go up, climb that window and come out from there. And when she is doing so, she is going to rub the body on the back of the stem node where stigma is there. Then she is going to rub the body on the anther and then come out. So if you look closely, it is still trying to come out, not reached here yet, actually. This was compiled from the pictures. I was holding a telephoto lens, 400 millimeter, because I didn't want to go close to the flower. So you can see some of the legs of the insects are appearing. And it comes out, and that yellow thing is the anther there on the back of staminode. She is struggling a lot. She has already struggled a lot, and she is struggling. Finally, she is out. So what is happening is the insect, the queen comes and probes on the flower. She is attracted by the color. So she comes for the color, then she is attracted by the fragrance. And she knows that the fragrance is coming out from here. So she is looking for food. She correlates fragrance with food and color also with food. So she is trying to look for how to enter through this so that she can get the food actually. Then she is trying a lot by facing the flower. Then she uses her brain and turns to one side. She knows that it cannot do this way, so she has to go sideways. And when she goes sideways, it is very fast, like flip, it goes in. Kind of it is pulled inside, and then she is trapped again because there is no food there, and she is not happy about it, that she has been deceived. So she is struggling to, she has no idea what to do, but she knows, she can see the window above, which is here on the back of the staminode. So she tries to climb up, and then she climbs up successfully, comes out, and while coming out, her back is rubbed on the anther, and she's out some pollen. Pollen is powdery, so you can see some pollen on the back of the body, and then she flies out. So if you look at this, I kind of expanded the time scale, and this is a 3-minute scale, and the pollinator landed on the flower here and went out of the flower here. This is around 2-minute duration, almost 10:45, 38, 10:47, 34, almost 2 minutes. So my observation says that around around 90 seconds to 120 seconds it takes to go in and come to the flower and then successfully pollinate. So it lands on the flower, then it turns sideways. It took 1 minute for her to think this. But the point is, if she was not able to enter, why was she trying so hard? So we will talk about that later on. Once she has entered, she is deceived, she is trying to get out of here, the only way is up, so she has to come out from there. So this is the structure we were talking about before, and this is in slightly in more detail. When the orifice, the rim of the orifice is bent, it is not touching the wall on the back. It is long, it is wide, but it is not touching the wall. And so the back side is slightly open. So it can see that it can move ahead, but it cannot move back. So the question third is what attracts pollinators to the flower, and what attracts pollinator to the front opening of the flower. Pollinator comes to the flower because of the bright pink color, but why it is planning to enter through the orifice? What is there? So we will talk about that later on. Here is the structure of the staminode, how it looks from front with 2 anthers on the back. This is the side view, and if you look from the inner view, you have stigma here. There is stigma here on the lower side, and then 2 anthers. The pollen is granular, a bit powdery. So this is the section of the pouch of the flower. The insect is going to come to the flower. It is going to enter the flower here, and it comes out from here. There is no other way out for her. In other species, I was in Hong Kong before, so there is Paphiopedilum purpuratum. In Paphiopedilum purpuratum, insects have been found dead inside the pouch. So it can happen. It can happen that the insect cannot find the way out and it dies inside. In this species, I have not seen, but it happens in this group. So next question is, after being deceived, the insect is deceived. After being deceived, why is it coming back to flower? Will she come back to the flower or not? So here are the 5 questions which originates from this study, and I'm going to address them one by one. What triggers the flowering? If you look at the time scale, the winter has snow, so this is the time when the temperature is low and humidity is very high. This habitat where this plant is found, the humidity is very high during winters. And the triggering point is when the temperature starts to rise. When the temperature starts to rise, the plant is going to emerge. And that is the stress call when it is going to produce the flower. So when we look at another second question, how will the species respond to climate change? So what if there is no snow? If there is no snow, the first thing is plant may not flower. Even if there is stress, even if the plant produces the green leaves, it may not flower. The flowering time may not match the insects. If there is no snow, then insect might have already come out and filled up her belly and went back to her nest. So the time of insect will not match. And if unfertilized flower maintained for long duration, then the plant health goes down. So the plant invests a lot of energy in producing flowers, then a lot of energy to keep it open. If the plant has to keep the open flower for 2 or 3 years, the plant is not going to survive. It is going to die. That is why some of the plants, they flower alternate years. First year, if they have flower, and if they are pollinated, they have to invest more energy. So next year, they will just have green leaves so that they can regain back the energy. So I have 3 hypotheses that the plant may not flower, flowering time may not match the insect, or unfertilized flower, if maintained for long duration, the plant health will go down. Then the third question, what attracts pollinators to the flower? So the bee, queen, she forages at low level. She can see the flower, which is almost this size. It is a large flower. It is visible from a distance if you are looking for it. It is so bright pink color. So she can see the flower large, and once she reaches the flower, she can see the opening of the front of the flower. And there are studies which says that if the flower is single colored and if the flower is multicolored, then the multicolored flower, they attract more pollinators. So the flower is veined. It has dark colored veins, which gives a kind of vibrant presentation in front of the pollinator. So it gets attracted to it. First attraction is based on the color. Second attraction is based on the fragrance. So what happens here? This is the section of the pouch. You have the pouch has pink color, which is attractive to the insects because the insects are coming to the flower because of this color. Then on the inner side, on the inner wall, my hypothesis is all the fragrance glands are located inside the flower. The insect doesn't sit outside the pouch on the other side. It stays on the mouth of the flower, the orifice of the flower. So the fragrance glands are located inside or maybe here and on the back wall. So the insect is here, and then what attracts pollinators to the front opening? There is fragrance, there is no reward. But the insect comes to the front. The fragrance glands, because they are present inside the pouch, what happens is during warm temperature, that pouch itself acts like a greenhouse. It becomes warm inside. When it becomes warm, it is kind of pushing the fragrance out from the orifice. So the insect can smell that, sense that fragrance that it is coming out from there, and at least it is stronger there. So if it is stronger there, something more is inside. Some food is there inside. The insect is relating it with the food. So during warm summer, because of the sunlight which falls on the pouch, the habitat is not very shady during flowering time. During flowering time, the trees are getting their leaves. It has not completely unfurled all the leaves. It is leafy, but not very dense. So sunlight is falling on the ground, and enough sunlight to make this chamber warm like greenhouse. And because the glands are located inside, they push the fragrance from the front, and that is the reason why insect goes there, because it is relating the fragrance with the food. So then after being deceived, this is very interesting. After being deceived, will the pollinator visit the flower again? There are some studies which says that the plant has very low fruit set because the insects, after getting deceived, they do not come back. Maybe so. Maybe they will not come back, and that is what is leading to fruit set. I'm not sure. Maybe yes, because so the insect is not looking at the complex structure of the flower. Insect is looking at the pink color and the fragrance. If the insect is roaming around and it finds another pink flower which has fragrance and has reward, then insect may also think that I missed something. I'm going to go and look back. It is not that it is going back to the same flower, but it will not think that all pink flowers don't have food inside. So once it is rewarded, it forgets that it was deceived before. And then third hypothesis is, does it matter? That's why I said bumblebees has a lifespan for 1 year. After 1 year, there will be another bumblebee. Even if he doesn't go to the flower again, next year there will be another bumblebee. Then there are other studies which shows that the insect has short-term memory. Short-term memory, it cannot remember anything for more than 1 day. What it did today, tomorrow it has forgotten. So this is a very nice aspect which can be studied. So this is my hypothesis. So to conclude, there is nothing I can conclude. It is the beginning of the study. So I would rather say to begin with, we have to look into the true distribution range of this species. We know the pollinator, but how pollination works, it is very important. And we need to study the pollination ecology of the species and how the plant is going to react with changing climate. This becomes very important if we are looking for the conservation of this species. And if we have all those data, then we can revise the conservation assessment, because even in Canada, I think it has lost half of its population. The pollinator has already lost 14% of its population in central USA. So it is concerning. If there is no pollinator, even if the plant is there, it is not going to help. And all this information can be later compiled into a species action plan. So this is the last slide of my presentation. And I'm really thankful to FIU for giving me this opportunity to be here and talk about part of my research. I'm thankful to Professor Hong Liu for giving this idea to have this talk. I'm thankful to Professor Shinto for organizing this, and Ms. Carey for organizing all the events. Holly and Mary, she took us, me and my wife and I, she took both of us around showing different labs, and I'm grateful for that and grateful for organizing this. I already have permission to collect samples. So if anyone wants to do research, I have 1 year permission, so I can still collect samples next year. I'm thankful to my professor at MDC College for encouraging me to do this. Jason Stern is one of the colleagues of my wife. Actually, the first flowering plant, we went together hiking looking for plant. The first flowering plant was found by Jason, so I thank him. And I thank my wife for bearing my tantrums and taking me wherever I wanted to go during all this. Not just this research, any research which I'm trying to do in USA. So this study and further plans are open for question if you have any questions. And if you want, you can have your student work on those aspects. Some of those are really very interesting. It needs an integrated approach, and it can give out some very good results if you want to take this study further. And if you have any questions, you can talk to me now, or you can email me at this address later on. Thank you so much.